It is about Huntington disease. Huntington disease, which is a disease caused by a abnormal expansion of DNA repeats, and that DNA repeat is CAG. CAG. This repeat base sequence is repeated in the Huntington's gene, HTT gene, and it was first identified in the year 1993. So there is a news about the Huntington disease. Research offers new hope for this disease treatment, and it is a uh, rarer than Alzheimer disease. Huntington disease has profound social impact because it is trying during the raising years. It means it affect the people's in between the age 30 to 50 years. So Huntington disease is a progressive and inherited. It is a inheritable neurological disorder. Has been regarded as previously it is regarded as incurable. So Huntington disease is caused by abnormal expansion of a base sequence, and that base sequence is CAG base sequence. This DNA repeats in the HTT genes, Huntington disease gene, and it was first identified in 1993, and it was considered that it is incurable. So healthy individuals have few repeats of CAG, but when the patient is suffering from this disease, it has a more than 39 repeats of CAG DNA base sequence. So longer repeats are associated with earlier symptoms. The patient showing the symptoms beyond inheritance, the repeat length can further expand in certain cell over a lifetime, and a process known as a somatic expansion, because it is transmitted not in the germ cell, it is transmitted through the somatic expansion, body cell expansion, which influence how rapidly the disease progress in that individual. So research has shown that Huntington disease mostly affect the patient's brain, and the brain changes occur decades before visible movements problem occurs. It affect the patient before visible movement, any visible movement problems occur. So the earlier damage, the first damage is seen in the brain part, and that brain part is striatum. The brain part is striatum. If the CG repeat is more than 39, it affect the brain part striatum, and a brain region which is striatum is important for motor control, where specific neurons gradually die in this patient. So as the disease advances, the degeneration is spread from the striatum to the cortex and white matter of brain, and affecting that patient cognition and emotional processing. Subtle impairments in attention, mood change also, and there is a change in cognitive behavior also. Cognitive flexibility have been detected many years before any diagnosis. So striatum is our corpus striatum is a cluster of interconnected. It is a cluster of interconnected nuclei that make up a large structure of the subcortical basal ganglia. So striatum is important for component of motor and reward system. So in this Huntington disease, the CAG repeat is more than 39. It affects the striatum of brain, which is important for motor and reward system. So functionally, the function of the striatum is to cognition, motor and action planning, decision making, motivation, reinforcement, and reward perception. When a patient is suffering from this disease, it will affect the cognition and behavior issues also. So how does Huntington disease is treated? So Huntington mutation occur in the DNA, and the sequence, the repeat is the CAG repeat. It occurs more than 39 times. So Huntington DNA is treated with the help of gene therapy and gene silencing method, which is also known as the RNA interference technology. So it is treated with the help of gene therapy. The infected gene is treated with the healthy gene. So because of this gene therapy, Huntington disease is treated. So Huntington disease is caused in an error in part of a DNA, and that Hunting gene is known as a Huntington genes, and error in CAG repeats is more. So if any parents of Huntington disease, there is a 50% chance that one parent is having Huntington disease. The 50% chance the offspring will receive that Huntington disease too. So these mutation turns a normal protein which is needed in the brain called Huntington proteins will be affected. So this Huntington protein permanently reduces, and the goal of the treatment is to reduce the level of toxic Huntington proteins permanently with the help of single dose. So therapy uses cutting edge genetic medicine. Combining this genetic medicine is combining gene therapy and gene silencing technology. So with the help of gene therapy method and then gene silencing method, gene silencing technology in which blocking the messenger RNA to transcribe. So so it is a breakthrough in gene therapy. It is medicine is taken with the help of the gene therapy, given with the help of gene therapy in which unhealthy HTT protein is recovered with the help of healthy gene of protein. So it is that gene is therapy is known as a MAT gene therapy, given is a one AMT 130. So it was a recent clinical work led by the researcher from University College London. So this therapy reduces the production of toxic HTT gene mutant. HTT mutant Huntington proteins reduces its protection with the help of this AMT 130. So a small trial involving clinically diagnosed protein participant, and this clinically healthy diagnosed protein is given in a small amount, which showed lower after this therapy, so lowered cognitive decline and also reduced levels of neurofilament light, which is a marker of neurodegeneration over 3 years. So this finding suggests that therapy may protect brain cell rather than merely easing symptoms. So with the help of this gene therapy AMT 130, it helps to reduce the mutant Huntington protein mutation, Huntington genes, and may protect the brain cell rather than merely treating only symptoms. Early detection and future treatment window are also theirs. So long-term studies of individual carrying the Huntington gene expansion conducts decades before expected any symptom is seen. So it has identified early marker of neurodegeneration and attentional deficits. So this finding indicate a potential treatment window long before motor symptom emerge. So it helping to know about the patient early before any motor symptom or any motor problem emerge in that specific patient. So a disease modifying therapies, gene therapy, gene silencing, with the help of that early intervention is possible. It could preserve cognitive function of that patient, improve quality of life, possibly extend lifespan of that patient. So it marks a significant shift in the outlook for people with Huntington disease. So Huntington disease is caused by repeat, and repeat is a CAG repeats, which is more than 39, leads to disease development. And it's the affected part of the brain is first is the striatum, then it moves to the cortex and white matter of the brain. So somatic expansion, body cell influences disease onset and progression, not germ cell. Huntington disease treatment market is there in 2024 US dollar 512.88 million is there. Key growth drivers are rising awareness and early diagnosis, technological advancement, increased research fundings. Germany market, United States markets, China markets, Saudi Arabia markets are making the medicine for this treatment of this disease, and leading companies are given Dr. Sedi, Shma, Any Lamps, Novartis, Sun, Hakma, Baus, Teva. These are the company which are making this medicine. So we have to know about the difference between the different types of neurodegenerative disorder which affecting the human primarily, that is affecting the brain of that patient. So its core symptom is there. Alzheimer disease meant the patient is associated with the memory loss, means decline in cognitive behavior. Parkinson, movement loss. Parkinson, motor skill is losing, means there is a loss of movements. And Huntington means it is an inheritable involuntary movement, plus there is a mental decline also. So Huntington is serious. That is, it affect the person mostly. So dementia is general umbrella terms used for the severe memory and cognitive issue. So Alzheimer disease is most type of dementia. So memory loss, cognitive decline, difficulty with daily tasks, And Alzheimer damage hippocampus, first memory of the brain. And the Parkinson disease, there is a movement disorders to tremor, stiffness, bradykinesia, slow movement, and balance issue are there in Parkinson disease. So damage dopamine producing cell in the brains. Parkinson, there is a dopamine producing hormone in the cell in the brain. Huntington disease, which is an inherited disorder, causes involuntary jerky movements, which is known as a chorea, psychiatric symptoms, cognitive decline. So it is caused by specific inherited gene mutation, HTT mutation, and the repeat is CAG. So dementia is umbrella terms which is used for overall term for decline in mental ability, including Alzheimer, vascular dementia, Lewy body dementia. Next news is about from environment. So Toby Kears win Tyler Prize for mycorrhiza research. American evolutionary biologist name is Toby Kears. She has been awarded the Tyler Prize for Environment Achievements, often referred to as a Nobel for the Environments. So Tyler Prize of Environment is known as is often considered as a Nobel Prize for the Environment, and it is given to Toby Kears. The award recognizes her Toby Kears' pioneering work in revealing in knowing the ecological and climatic importance of mycorrhizal algae. So he worked on mycorrhizal importance. What is the importance of mycorrhizal fungi in ecology and climatic importance? So here is worked in a expert based at a Breze University, Amsterdam, and she is the youngest woman to win this award, Tier Prize Award for Environment Achievements. So about the carbon in global mapping, how it is important, what is the role of the mycorrhizal fungi. So plant channels provides surplus carbon below ground, where mycorrhiza fungi, there is a presence of mycorrhiza fungi in the plants, roots of plants, store a estimated 13.12 billion tons of carbon dioxide annually, roughly one third of fossil fuel emission. So it helps in the absorption of this one third of fossil fuel carbon dioxide emission. Mycorrhizal fungi use this carbon below the ground. By co-developing a global underground atlas, Kears and his colleagues mapped the worldwide distribution of these mycorrhizal fungi, highlights the vast reservoir of below ground biodiversity. There is a presence of mycorrhizal fungi which help to to absorb the carbon below ground and utilize it. So this finding, which is harmful for the society and for the environment, so this finding provide critical data for environmental planning, especially as many fungal hotspots lie outside protected areas. So it is considered as a biological marketplace theory. In a landmark 2011 paper published in Science, Toby Kears explained that mycorrhizal fungi operate as a rational trader in a biological marketplace. So it worked as a rational traders in a biological marketplace, in biological place, through microscopic filaments present in this, with the help of microscopic scopic filaments present in mycorrhizal fungi. It down the root supplied phosphorus and nitrogen to plants in exchange for carbon-based sugar and fats. So it is a symbiotic association. It's mycorrhizal fungi take carbon-based sugar and fat for their growth, and its fungi provide phosphorus and nitrogen from the soil to the plants to the atmosphere. So in experiments, so fungi actively shift nutrients from areas of abundance below ground to scarcity to maximize carbon returns, mirroring supply and demand dynamics seen in human economics. So it is alarmed by conservation gap. Tears co-founded the Society for the Tears co-founded the Society for the Protection of Underground Networks. It means the underground below ground networks, to advocate legal protection for fungi, to how to protect the fungi because they are beneficial for environment. So they are symbiotic association, taking the carbon based sugar and fat for their growth, and then taking the phosphorus and nitrogen from the soil to the plants, providing to the plants. So mycorrhizal fungi are symbiotic soil fungi. Fungi that, because fungi do not photosynthesize, mycorrhizal fungi symbiotic association with the plant connect with the plant roots. So it create an extensive network that drastically increases water and nutrient, uptake, especially phosphorus and nitrogen, while receiving carbon-based energy from the plant. These fungi improve plant growth. It also provides resistance to different type of stress to plant and soil structures, benefiting over 80% of terrestrial plant species. So this is a plant with mycorrhizal fungi, how the development is take place, and this is the plant without mycorrhizal fungi. So this act as a biological marketplace. So the theory given by Years published research showing that mycorrhizal fungi behave like a traders, free traders, swapping nutrients, phosphorus and nitrogen from the soil for carbon-based sugar with plants based on supply and demand. So providing the phosphorus and nitrogen from the soil and taking carbon-based sugar for their growth. So in this way, symbiotic take place and act as a symbiotic association take place, act as a biological marketplace. So mapping network also, and this uses innovative technology with the help of robotics to visualize and map global mycorrhizal biodiversity. Most of the vital networks are not located in protected areas. Robotics are not located in that but protected areas. Now, key achievements is the climate action. It's help in this. He work highlight that mycorrhizal fungi networks are important for climate regulation, with plants allocating an estimated 30 billion tons of carbon dioxide annually to these subterranean networks. So, as co-founder of the society, as Kier become the co-founder of the Society of for the Protection of Underground Network, SPUN. Kier becoming as a co-founder of Society for the Protection of Underground Network, SPUN, she works to turn her research into direct environment policy and legal protection for fungal habitats, and in this way, in turn, it protect the plant growth also and protect the environment as well. So important point is, a mycorrhizal fungi form symbiotic network with plant roots. They sequester over 30 billion tons CO2 annually. So in this way, CO2 is utilized and protect the environment, save the environment. The Taylor Prize include a momentary award. The amount is two lakh 50,000. So most fungal biodiversity hotspot lie outside protected areas. Next update is about from the Science Communication Institute of Science Communication, CSIR, National Institute of Science Communication and Policy Research, CSIR NISCPR, celebrated its fifth foundation day on January 14, 2026 in New Delhi. So these events highlighted 5 years of progress in science communication, policy research, and the validation of traditional knowledge utilized in science. So the key highlights of this fifth Foundation Day, CSIR and NISCPR, launch of portal, and the portal name is Swastik Portal. A major highlight was launch of Swastik Portal. The full form of Swastik is scientifically validated societal traditional knowledge web portal is launched. This platform aims to to provide scientific evidence on traditional knowledge in English. Included 19 Indian languages and 5 foreign languages also. It also focuses on artificial intelligence and science communication to make science accessible to a broader, more diverse audience. It also helps in digitization of traditional knowledge, and it highlighted the full digitization of TKDL, Traditional Knowledge Digital Library, and its importance in global validation of traditional knowledge. So, key dignitaries in the fifth Foundation Day was chief guest for the event was Dr. Shiv Kumar uh, Kalyanaraman, CEO of the Anusandhan National Research Foundation, ANRF. The event was led by Dr. Geeta Vani Rayasa, Director of CSIR and NISC PR. So the key dignitaries are CEO of Anusandhan National Research Foundation, Dr. Shiv Kumar, and director is Dr. Geeta Vani. So these are the key dignitaries. In this, the scientific recognition is given by they showcase the poster from PhD scholars and project staff, underlying the institute's commitment to bridging scientific research with societal needs. So CSIR and ICPR was established in the year 2000. It is established in the year 2011. I mean, it has to my merging with the help by merging two laboratories, CSIR National Institute of Science Communication Information Resource, and second one is the CSIR National Institute of Science Technology and Development Studies. So launch of Swastik and academic engagement platform take place. So Shiv Kumar is the CEO of the Anusandhan National Research Foundation. He praised NISCPR's alignment with ANRF vision of positioning India as a global research and innovation hub. On the occasion, Dr. Kalyanaraman launched the Swastik web portal, a national initiative implemented by CSIR NISPR to disseminate scientifically validated traditional language. The Swastik content is available in English, 19 Indian language, 5 foreign languages, enhancing accessibility and outreach. So important fact is it focuses on science, communication, policy research, and traditional knowledge. TKDL is a globally recognized digital depository of India's traditional knowledge. ANRF aims to catalyze India's rise as a research of innovation powerhouse. And AI is being explored for regional language, science, communication. Next update is scientists develop low-calorie natural sugar tagatose. It is a very good news for the diabetic patients. So there is a discovery of low-calorie natural sugar, and the name of that sugar is natural sugar is tagatose. So scientists at Tufts University has developed a very high efficient, cost-effective method to mass produce tagatose. So tagatose is a rare natural sugar. It is a rare natural sugar. It provides sweetness of like a table sugar with about one third of the calories. So it has only one third of the calories, and tagato is a rare natural sugar that offer a taste of regular sugar without causing any sharp spike and increase in insulin levels. So this development could pave the way for healthier sweetening option and reduce dependence on artificial sweeteners. So it is a very good news for the diabetic patients. So the breakthrough production method is historically, as we know, that tagatose has been expensive to produce because it only occurs in very small amounts in dairy and some fruits. Tagatose present in trace amount in dairy and some fruits. So the new technique published by Cell Reports Physical Science in early 2026, it helps in transforming common glucose. It helps in developing, transforming common glucose into tagatose using with the help of engineered bacteria. And that bacteria is Escherichia coli. So engineered Escherichia coli is used to act as a tiny factories for the production of natural sugar tagatose. And there is also they use novel slime mold enzyme also. They inserted a specific enzyme from a slime mold. The enzyme slime mold name is a Dictyostelium discodium. So they inserted a specific enzyme from a slime mold, Dictyostelium discodium, called the enzyme name is galactose 1 phosphate selective phosphatase, GAL1P. So they have inserted this enzyme also from slime mold to help in the production of tagatose. So this process achieve production which yields up to 95%, far suppressing industrial method that range from 40 to 77%. So it's give rise to 95% of tagatose production. There are many key benefits of tagatose. It acts as a bulk sweetener, meaning it has the physical properties as of real sugar. So authentic taste and texture. It approximately 92% as sweet as table sugar, which consists of sucrose. Unlike many artificial sweeteners, it browns and caramelizes during baking. Now it's diabetes friendly. It is tooth friendly also. It inhibits the growth of harmful oral bacteria, and it acts as a prebiotic effects also. Tagatose. It is only partially absorbed in the small intestine. The rest is fermented in the colon of large intestine, where it supports gut health by acting as a prebiotic. So it's very important. Authentic taste and texture, diabetic friendly, tooth friendly, prebiotic effects. Now, safety and availability, the US Food and Drug Administration, FDA and WHO has already designated tagatose as genetically recognized as safe. GRA genetically recognized as safe, while currently available in some specific specialty products on sites like it is present, like some website like Amazon or Desert Cart. This new production method is expected to significantly lower prices and increase its presence in mainstream grocery items. So tagatose and why it matters. Tagatose is about 92% as sweet as sucrose, but contain only one third of the calories. So calories only one third. So tagatose has a minimal impact on blood glucose level. So there is a little with diabetes or insulin resistance. So people is suitable for the diabetes or insulin resistance patient. Tooth friendly, health friendly, harmful oral bacteria act as a prebiotics. So breakthrough in it is a breakthrough in large scale production. Researcher from the Tufts University in collaboration with biotechnology firms, and that firm's name is Manus Bio and KCAT Enzymatic, have developed a highly efficient method to produce tagatose at a scale. So University in collaboration with biotechnology firms Manus Bio and KCAT Enzymatic have developed this tagatose at large scale, and their findings were published in Cell Reports Physical Science. So they engineered bacteria as sugar factory. That bacteria is Escherichia coli to act as a microscopic production units, and they also identify a new enzyme from slime mold, lactose 1 phosphate selective, introduced to convert glucose into tagatose. So these are the method. So it give up to 25%, far exceeding current commercial techniques. So it is about 92% as sweet as sucrose. Engineered E. coli bacteria enabled 95% production yield. The research was published in Cell Report Physical Science, and it is a low calorie natural sugar, not artificial sugar, with minimal insulin response. Next update is wolf puppy meal reveals woolly rhinoceros genome. Wolf puppy meal reveals woolly rhinoceros genomes. From we get the woolly rhinoceros genome we get from the wool puppy meal. So a 14,000-year-old weak wool puppy frozen in Siberian permafrost has revealed a complete woolly rhinoceros genome from its last meal, making the first time a genome was retrieved from another animal stock. So it was first time happened that any genome is obtaining one genome is obtaining from different type of animal's stomach. So it is the first time happened. So this analysis indicate the species name is Coelodonta antiquatis. Coelodonta antiquatis was not heavily inbreed and suffered a rapid climate driven population collapse rather than a long decline. So a 14,000-year-old wool puppy frozen, and it was frozen in Siberian permafrost, and it has revealed complete woolly rhinoceros genome from its last meal. So it is a unique discovery. The remains were found in 2011 near the Siberian village of Tumat. The wolf puppy was only a few weeks old and likely fed by its mother. So it's the genome analysis. Researchers recovered high quality DNA from the undigested rhino meat, providing insight into the species' final days. So there is a no long-term inbreeding. So contrary to theories of slow decline, the genome showed the woolly rhino population was genetically stable and healthy until very close to its extinction. So the findings support that the rapid climate warming events rather than the overhunting by humans caused their extinction around 14,000 years ago. So it was expected that because of the overhunting by humans, their extinction take place. But the finding demonstrate that wolf during these periods, or at least the parent of this puppy, fed on rhinoceros carcasses. So this breakthrough study was published on January 14 in Genome Biology and Evolution. It was a unique discovery. So wolf puppy meals reveals woolly rhinoceros genome. So genome recovered from permafrost discovery with the help of the permafrost preservation, which involves a ground temperature is below 0 degrees Celsius to prevent the release of trapped greenhouse gases, protect ecosystem, stabilize infrastructure. So permafrost means temperature below 0 degrees Celsius. So researchers extracted DNA from chunk of undigested rhinoceros meat found in the stomach of the mummified wolf puppy. So they extract the DNA from chunk of undigested rhinoceros meat, and this meat found in the stomach of mummified wolf puppy. So this remarkable preserved material enabled scientists to reconstruct a highly quality genome of woolly rhinoceros. Woolly rhinoceros is a very cold adapted herbivore that one roamed northern Europe and Asia. So this finding shed new light on the biology and extinction dynamics of the iconic Ice Age species. So important fact is the woolly rhinoceros species name is Silodonta antiquitatis. It went extinct around the age of last ice age, about 14,000 years ago, but its genome recovered. Genome recovery was possible due to permafrost technology. So rapid climate warming caused loss of steppe tundra habitat. So it's in this finding is that it's a unique discovery. The wool puppy mill reveals woolly rhinoceros genome. Next update is CIMFR conducts India first hydrogen compressor safety assessment. So it is also boon for the environment. India has marked a key milestone in clean energy production, clean energy safety with the country's first comprehensive safety assessment of a hydrogen compressor dispenser system. Production of India's first hydrogen compressor dispenser system, and this assessment was conducted by Jharkhand Dhanbad-based CSIR CMF FR, which reinforcing India's preparedness for scaling up hydrogen infrastructure, clean metal infrastructures under the natural hydrogen mission. So facility location and inauguration plan, the facilities installed at the Netra campus of NTPC Limited in Noida, Uttar Pradesh. So, according to the official, this installation is scheduled to be inaugurated by Prime Minister, and the initiative is expected to significantly strengthen the operational framework for hydrogen refueling infrastructure in the country. So it will accelerate the rollout of hydrogen powered vehicles and refueling station, and in turn it will protect the environment because there is a less emission by hydrogen powered vehicles. So this development is expected to reduce dependence on fossil fuels like carbon, petroleum, natural gas, and contribute to lowering less carbon emission across India's energy and transport sector. So in turns, it will help in improving the atmosphere and environment, reducing less emission of carbon emission. So CSIR, CIMFR function under the Council of Scientific and Industrial Research. Gas group 3rd C represent highly explosive industrial environments. This facility supports India's national hydrogen mission objective for cleaner fuels. So this critical evaluation ensures safe high pressure operation in high explosive gas Group third C environment, marking as a key milestone in India's national hydrogen mission. So its significance, it is the first full system assessment important from green hydrogen, clean hydrogen fuel infrastructure, and expert examines flame proof equipment, intrinsically safety system, mechanical components, safety mechanism, and location and scope conducted at the NETRA campus. It evaluates the system used for fueling hydrogen vehicles to support the transition from fossil fuel as highlighted in the Facebook post also, and it becomes India become able to produce a cleaner fuel instead of using fossil fuel. So the certification ensures safe reliable operation of hydrogen refueling station. So led by Dr. AP Singh and directed by Professor AK Misra, this achievement by the FLP division of CSIR CIMFR strengthened India's hydrogen infrastructure safety. Next update is India scientists develop artificial intelligence tool to decode disordered proteins. Disordered proteins. So researchers at the National Center for Biological Science, NCBS, Bengaluru. So this is news from Bengaluru. So National Center for Biological Science, Bengaluru researchers have developed disobined, disobined DSOBIND is an open source deep learning tool. It is an open source deep learning tool that decodes, that help to know about the how intrinsically disordered protein binds to their neighbor proteins or bind to its partner. So these tool help to know about the intrinsically disordered proteins in your body, how it binds to the partner proteins and create problem to the patient. So these sharp shifting flexible proteins, intrinsic binding protein are important for cell signaling, but often defy traditional structure analysis. So disobin tool uses protein language models. It uses protein language because it has to know about that intrinsically disordered protein. So it uses protein language model to identify intrinsically disordered proteins interaction, accelerating research into cancer. And because of this disordered proteins, the patient is suffering from different types of cancer. The patient is having different types of neurodegenerative diseases, and therapeutic design is also required. So key details about this OBINS, which is a open source deep learning tool, and target is to focus on increasingly disorder reason of these or proteins that lack a fixed structure, because as the word suggests, disorder protein, so it lack fixed structure, often called disordered protein. So safe shifting proteins. So this is the reason the tool is known as a disobedient. So it functions to know about the binding site and its interaction, acting as a partner aware tool, meaning it analyzes the interaction between disordered protein and its binding partner, and it's acting on to know about the different types of problem the patient is facing, whether it is accelerating into cancer, neurogenetic disorder, etc. So it uses deep learning models trained on vast protein sequence data sets rather than depending only on traditional 3D structured mapping. So this OBIN tool application in understanding different types of cancer, neurodegenerative diseases, and cell communication. So it is available for global scientific use. It is open access, help in precision medicine and cellular studies, developed at NCBS Bangalore. So Tata Institute of Fundamental Research was supported by funding. The funding support is given by Tata Institute of Funding Research from the Department of Atomic Energy, Department of Science and Technology, SCRB, and Department of Biotechnology. These obedient protein AI tools for predicting IDP interactions. So intrinsically disordered protein lack signal 3D structure, flexible disobedient deep learning tool are used to know about the no prior structure information needed. So predict binding to partners and how it's causing the problem like cancer and neurodegenerative diseases. So it performance and uses are disobedience is about the use alongside for higher accuracy disobedience plus alpha fold Meyer is used its application in immune signaling, cancer research, and neurodegeneration problems. So future impacts its help in knowing about the different to developing about the different types of therapeutic medicine. So, the question may arise, why intrinsically disorder protein matter? Why it is important? Because unlike most protein, as we know about the most protein, fold into stable three dimensional structure. So, protein is a building block of our life, and most proteins always fold in stable three dimensional structure. But about the intrinsically disorder proteins, it lacks a fixed structure, fixed shaped. So, these shape shifting molecules are central and lead to cellular life. They regulate signaling networks, control gene expression, help protein folding and quality control, and help from dynamic cellular structure known as condensate. So their flexibility, their changing shape, while biologically advantages has made them difficult to analyze to know using conventional structure methods. So the disobedient technology deep learning tool is developed to, and this newly developed tool named disobedient analyzes these changing protein shape sequences to know about which region of a disorder protein will interact with a binding partner. So it uses protein language model, and model is a protein language to know about the disarranged protein. So it uses protein sequences to predict which region of the disorder protein will interact with the binding partners. It uses protein language models. So a form of protein language model is a type of AI trained on million of known protein sequences. Crucially, this OBIN does not require prior structural information, previous structural information, or sequence alignments, and explicitly account for the binding partner, a key factor in intrinsically disorder protein interactions. So important factor for exam is intrinsically disorder protein lack a fixed stable 3D structure. Protein language models or AI system trained on large protein sequence data set, and that tool is a tool is disobedience. NCBS is a premier life science research institute under TIFR. So National Center for Biological Science, NCBS, is a premier research institute in India, which was established in the year 1991 with function as a constitute center for Tata Institute of Fundamental Research. TIFR, it is situated in Bengaluru, Karnataka. Main campus in Gandhi Krishi Vigyan Kendra. AlphaFold tools are also used to know about the protein structure and interaction prediction. So another tool is the AlphaFold tools. So about what is intrinsically disorder protein? Intrinsically disorder proteins are reason are functional proteins that lack a fixed stable 3D structure. It lack a fixed stable 3D structures. So under physiological condition, under any physiological condition, it lacks a fixed structure. So instead, they exist as dynamic ensembles of rapidly changing shape and conformation. They are rich in specific polar and charged amino acid with low hydrophobicity. So they act as a flexible linker. Signaling molecules are functional copyrions, which is also a type of protein. So IDPP are often described as having high stability despite their structural instability. It is also considered as having high stability despite their structural instability, which is known as a stability of instability paradox. It is a one type of stability of instability paradox. So highly this intrinsically disordered protein present in we humans, animals like eukaryotes, they consequently roughly 30 to 40% of the proteomes, and they are involved in different types of disease, malfunctioning or misfolding happen in this IDBPs, intrinsically disorder proteins linked to neurodegenerative condition like Alzheimer, Parkinson disease, due to their tendency to clump together. Next update is Nagaland Beijing study turns apple leaves into corrosion shield. So it's a very big achievement that Nagaland and Beijing Indo-China study turns apple leaves into corrosion shield. So a joint research collaboration took place between India and Chinese university to protect metal from different types of corrosion, using with the help of to protect metals with the help of waste apple leaves. So they have used waste apple leaves to protect metal from corrosion using waste apple leaves, and collaboration is done by India and Chinese. So this breakthrough offers an eco-friendly alternative to toxic industrial inhibitors and highlight the growing role of green chemistry in infrastructure and manufacturing. So collaborative study of India, Nagaland University and China University of Science and Technology, Beijing has developed a green corrosion inhibitor, and this green corrosion inhibitor is developed with the help of waste apple leaves to protect metal from corrosion. So the researchers converted these agricultural waste apple leaves into carbon quantum dots, quantum develop into quantum dot ACDS, carbon quantum dots. They create a protective layer above the metal, shielding copper from acid corrosion with over 94 to 96% efficiency. So this sustainable non-toxic alternative promises to reduce industrial reliance on hazardous chemical which is released by the industry. So protecting the industries while combating costly metal oxidation. So key detail of the study is it is led by Professor Ambrish Singh, Nagaland University, and Professor Yuzi Huang, University of Science and Technology, Beijing. Process in this apple leaf waste was transformed into nitrogen and sulfur doped carbon quantum dots. So in this we have protecting the metal from corrosion by using apple leaf waste, and it is transformed into nitrogen and sulfur dose carbon quantum dots SCDS with the help of hydrothermal process. So the ACD coating forms a compact stable film on layer or film on copper surfaces, preventing ion exchange and oxidation so that no corrosion take place, with inhibition efficiency reaching 96.2% over longer exposure. So it's a big achievement. So this eco-friendly waste to health method offer a sustainable alternative for industries like oil and gas, manufacturing and power generation. So they have created waste to wealth and act as a help in green chemistry. So the finding were published in the Journal of Alloys and Compounds. The team plans to move from laboratory scale validation to pilot. So it is the Indo-China academic collaboration. Indo-China academic collaboration led by Professor Ambrish Singh of India and Professor Yuzi Kwang of Beijing's instead of discarding apple leaves, they have used apple leaves into high performance corrosion, act as a high performance corrosion inhibitors, and it can address global industrial challenges through sustainable science. So apple leaf carbon quantum dots explains. So researcher developed carbon quantum dots derived from apple leaves using a with the help of green hydrothermal process, and these nanoscale particles doped with nitrogen and sulfur create multiple active sites that strongly bind to metal surface, attach to metal surface. Laboratory tests showed that these apple leaf carbon quantum dots, when attached to metal surface, can suppress copper corrosion in acidic environments with inhibition efficiency ranging from 94% to 96%. So it is industry. There is a industrial. It is important for industrial environment also. So corrosion is a persistent problem, regular problem in sectors like oil and gas, power generation, chemistry processing, and wastewater treatment, where metals are exposed to harsh acidic condition. So all these industries are metal are exposed to harsh acidic condition, and they are tend to more corroded. So the study found that bio-derived inhibitors with the help of waste apple leaves form a compact and stable protective film layer on copper, blocking corrosive ion transfer and extend the service life of pipelines. It help in improving the service line of different types of pipelines. storage tanks, industrial equipments, while reducing environment and health risk, which is linked to conventional chemical inhibitors. So carbon quantum dots are nanoscale carbon-based material with functional surface groups. Corrosion inhibitors reduce metal to degrade in acidic or harsh environment. So it is a one type of waste to wealth approaches which supports circular economy models and also protects environment. So green chemistry is boosting, it is focusing on reducing toxic substances in industry processes. Next update is about Guillain-Barré syndrome outbreak in Numuch district. So a Guillain-Barré syndrome's outbreak took place in Madhya Pradesh, and this district was Numuch district's Manasa area. Has led to two reported death. Two deaths took place and at least 18 cases, 9 confirmed as of January 2026. It is an autoimmune neurological disorder. It is also very serious type of disorder because it is autoimmune, which is in autoimmune means a healthy cell is attacking its own cell. So autoimmune neurological disorder affecting brains, which cause severe symptoms, prompting health teams to conduct door-to-door screening and set up dedicated treatment wards with free government coverage took place in Madhya Pradesh. So it was seen in Manasa town, Nimuch district of Madhya Pradesh. Two children reported dead with over a dozen cases under treatments. 18 total cases. 15 teams conducted door-to-door screening about the Guillain-Barré syndromes. It is a rare autoimmune disorder where the immune system attack the peripheral nerves, which is leading to weakness and potential paralysis. It causes potential paralysis. It affects peripheral nerves. So symptoms rapidly progressing muscle weakness, numbness, starting from your leg, from starting in limbs, as explained in La Excellence IS Academy post. So it is not contagious, it is not communicable. It is often triggered by preceding bacteria, by bacteria or viral infection, and the virus is Campylobacter jejuni. Authorities in Western Madhya Pradesh, Nimuch district are on the high alert following a certain outbreak of Guillain-Barré syndromes. It is a rare immune-mediated neurological disorder. So Guillain-Barré syndrome is a rare but serious autoimmune disorder because the healthy cell is affecting the uh, uh, own cell, so in which body own cell in which the body immune system mistakenly attack the peripheral nervous system. The system includes the nerves outside the brain and spinal cord, so which control muscle movement and transmit sensory information. So Guillain-Barré syndrome is an autoimmune serious disorder affecting our peripheral nervous system. So it is often triggered by infections such as a virus, Campylobacter jejuni, influenza, viral illness. IVG and plasma exchange are the two main treatment for this disease. Most patients recover, but severe cases can cause respiratory failure. So, understanding Guillain-Barré syndrome's early sign: tingling in fingers, toes, muscle weakness in legs, progression weakness spread to arms, upper body difficulty walking, severe stage paralysis. Treatment option is plasma exchange, IVIG, and physical therapy. Recovery: most people regain full mobility with proper care, but in severe case it leads to respiratory problems severe. Webb telescope, next update is Webb telescope delivers sharpest dark matter map yet. So using observation with the help of James Webb telescope, the new map provides deeper and sharper insight information into how a galaxy formed and evolved during a critical phase of cosmic history. So cosmic history with the help of James Webb telescope, to know about the how galaxy formed and evolved during the critical phase of cosmic history, which is called cosmological evolution, is the scientific study of universe origin, development, and large scale structure from Big Bang to the present day. Big Bang is 13.8 billion years ago. So it maps key milestone like the formation of atom, star, galaxy, and accelerating expansion driven by dark energy. So the James Webb telescope has produced the sharpest and detailed matter to detailed map of dark matter to date, serving a significant portion of the sky, and that significant portion of the sky known as a cosmos field. So the map twice as sharp as previous Hubble maps. Hubble map uses data for 80,000 galaxies to trace the invisible cosmic wave or dark matter that act as a scaffolding for the universe, validating existing gravitational models. So, what are the new dark matter map? Key findings are: the study in Nature Astronomy notes that map offers over twice the resolution of the previous Hubble Space Telescope map. The map includes data from nearly 8 lakh galaxies in cosmos field, identifying far more galaxy than prior surveys. So this map shows the dark matter form a scaffolding or hidden skeleton for the universe, which directly influence the distribution of visible matter. Researchers created the map by studying how dark matter, gravity, gravity how dark matter's gravity wraps light from distant galaxies, a technique known as a weak gravitational lensing. So it's a key details finding a dark matter and regular matter clumped together. Findings show that large luminous galaxies are located in the center. Large luminous galaxy which are located in the center of the dark matter halos, affirming that dark matter and regular matter have always been the same place. Of affirmation is that dark matter and regular matter have always in the same place. So impact on it has impact on understanding of the universe, cosmic wave validation. The map reveals a complex web of dark matter, filaments and connection, which provides strong visual evidence for current cosmological theories of how the universe formed, how the universe evolved. It also suggests about void analysis. The map will be used to better understand galactic voids, a region where voids where few galaxies exist. It also gives future prospects, as the study led by the researcher like JPL astrophysicist Dianos Skogmiola suggests that this pave the way for deeper, more comprehensive mapping, especially with upcoming mission like Nano Grace Roman Space Telescope. Beyond earlier, it was seen by the Hubble-based map. Previous large-scale dark matter map totally depend on Hubble-escape telescope. Wave's infrared capability and greater light gathering power delivery nearly double the resolution. James Webb Telescope just nearly double the resolution capacity while also peering further back in time. So this the map looks 8 to 10 billion years into the past, which is a crucial era when galaxies were acting, forming and assembling. So dark matter constitute about 85%, the important point, 85% of the universe. Dark matter constitute. Weak gravitational lensing is used to map invisible mass. The James Webb Telescope helped to operate mainly in infrared wavelengths. It is mainly operating infrared wavelengths. The Cosmos Survey is a key field for studying large-scale cosmic structure. So, by locating dark matter halos, the cradle of galaxy, the finding place strong constant on model explaining how the universe evolved from an almost universe. So these are about the update. Now kindly watch the next series. Thank you for watching. Keep consistent, maintain consistency.